Today, we will explore the neuroscience of fear, including trauma and post-traumatic stress disorder, PTSD. Although the neuroscience of fear has deep roots in biology and psychology, advances over the past 10 years have revealed not only the neural circuits and brain areas that control the fear response but also effective methods to extinguish these fears through behavioral therapies, medications, and brain-machine interfaces. We will discuss all these aspects, providing a comprehensive understanding of the biology of fear and trauma, along with several practical tools. Prepare for an in-depth exploration of the biology of fear and trauma, and discover practical methods to confront and overcome them. Chapter 1: The Biology of Fear and Trauma. Fear. A short word, yet it carries immense weight. Over the centuries, this nervous system response has been one of the most studied emotions in the quest to understand how the body and mind prepare to face the unknown and the dangerous. Fear is not just a fleeting feeling, it manifests as a physiological response involving various changes in the body, from increased heart rate to altered blood flow. However, this reaction also has a complex cognitive component, filled with thoughts and memories that shape the perception of danger. To understand fear, it is essential to differentiate it from other related states, such as stress and anxiety. Stress is a physiological reaction that prepares the body to face immediate challenges, characterized by rapid breathing and a series of other bodily changes. Anxiety, often seen as nervous anticipation, is generally a concern about future events, a form of projected stress for what is to come. Fear, in turn, can include elements of both stress and anxiety but also manifests on its own, often suddenly and intensely. Fear can be seen as a natural alarm, an instinctive response to perceived threats. But what happens when that alarm doesn't turn off? This is where we enter the territory of trauma and post-traumatic stress disorder, PTSD. Trauma occurs when a fear experience is so intense that it is deeply embedded in the nervous system, reactivating at unexpected and often inappropriate moments. PTSD, in turn, involves traumatic responses that persist and are triggered by stimuli that, rationally, no longer represent a real threat. Imagine living with a constant sense of imminent danger, where a sound, a smell, or a sight can instantly transport the mind into a state of panic. The study of fear reveals that our reactions to threats are complex and deeply rooted in our biology. At the cellular level, fear involves an intricate network of neurons and synapses that form specific circuits in the brain. These connections are responsible for detecting, processing, and responding to threatening stimuli. Brain areas such as the amygdala and the hippocampus play crucial roles in this process. The amygdala, an almond-shaped structure located in the limbic system, is central to threat detection and generating emotional responses. The hippocampus, on the other hand, is involved in memory formation and contextualizing fear experiences, helping to distinguish between real threats and false alarms. But fear is not just a product of the brain. It involves the entire body, activating the autonomic nervous system and releasing hormones like adrenaline and cortisol. These substances prepare the organism for the fight-or-flight response, increasing vigilance, energy, and the ability to react quickly to imminent dangers. However, when this response becomes chronic, as in the case of PTSD, the body can suffer negative consequences, such as an increased risk of cardiovascular disease, sleep disorders, and mental health problems. The relationship between fear and trauma is a fascinating and challenging field of study. Trauma, unlike momentary fear, represents a lasting mark on the nervous system. When a person experiences an extremely stressful event, such as an accident, an assault, or a natural disaster, the brain can enter a state of hyperactivation, where the amygdala remains on high alert. This hyperactivation can make it difficult to distinguish between safe and threatening situations, leading to exaggerated reactivity to harmless stimuli. PTSD is an extreme manifestation of this condition, where individuals relive the traumatic event through flashbacks, nightmares, and a persistent sense of anxiety. The brain of someone with PTSD is in a constant state of vigilance, as if always waiting for the next danger. This not only affects mental health but also has profound implications for physical, social, and emotional well-being. The biology of fear and trauma also involves how these states can be unlearned or extinguished. The extinction of fear is a process by which the brain learns to dissociate a previously threatening stimulus from a fear response. This process is not simply the elimination of fear but a reconfiguration of neural associations. In other words, to overcome fear, it is necessary to create new memories that replace old threatening associations with positive experiences. For example, a person who is afraid of dogs due to a past bite may begin to overcome this fear by gradually interacting with friendly dogs, forming new memories that weaken the association between dogs and danger. However, this replacement of memories is not a simple process and may require time, patience, and often specific therapeutic interventions. Behavioral therapy, such as gradual and controlled exposure to feared stimuli, has proven effective in extinguishing fear. Additionally, advances in drug therapies, such as the use of beta blockers to reduce the physical symptoms of fear, offer new hope for those suffering from PTSD. Even more promising are brain-machine interfaces, emerging technologies that allow direct modulation of brain activity to reduce fear and trauma responses. A recent study shed light on an innovative approach to dealing with fear and trauma. Researchers discovered that 5 minutes of daily deliberate stress exposure can alleviate symptoms of depression and persistent fear. This study suggests that controlled stress exposure can help recalibrate the nervous system, making it less reactive to threatening stimuli. Although more research is needed to fully understand the mechanisms behind this discovery, it opens new perspectives for the treatment of conditions related to fear and trauma. The biology of fear and trauma is a rich and constantly evolving field, offering profound insights into how our bodies and minds respond to danger. Understanding these mechanisms not only helps us better cope with our own reactions to fear but also develop more effective interventions for those suffering from trauma and PTSD. The science of fear, by unraveling the mysteries of our most primal responses, illuminates paths to healing and resilience, showing that even in the shadows of trauma, there is always a light of hope. The journey to understanding fear and trauma is complex, filled with nuances and intricate interactions between body and mind. From the initial identification of danger to subsequent physiological reactions and lasting memories that shape our behavior, each aspect of this emotional response is a piece of a larger biological puzzle. Through continuous study and practical application of this knowledge, we move one step closer to understanding and, eventually, mastering fear, transforming it from an obstacle into an opportunity for growth and learning. Chapter 2: Neural Circuits of Fear. Within the labyrinth of the human brain, the amygdala emerges as a central figure in the fear response. This small cluster of nuclei is a vigilant guardian, always on the lookout for danger signals. When it detects a threat, it triggers a series of reactions involving both the body and mind, preparing us to face what is to come. The threat response, in essence, is an intricate dance of neural and hormonal signals that mobilize all necessary resources to ensure our survival. The threat reflex, mediated by the amygdala, is an almost instantaneous process. As soon as a threat is perceived, the amygdala activates systems that increase vigilance and suppress calming responses. This activation is crucial to ensure we are ready to react quickly. The body enters a state of maximum alertness, where every sound, every movement is meticulously monitored. In this state, the mind becomes an tireless sentinel, focused exclusively on survival. The HPA axis, which includes the hypothalamus, pituitary gland, and adrenals, plays a vital role in this process. This axis is responsible for releasing hormones like cortisol and adrenaline, substances that flood the body and prepare it for action. The hypothalamus, upon detecting amygdala activation, signals the pituitary to release hormones that, in turn, instruct the adrenals to release cortisol and adrenaline. This hormonal flow increases heart rate, redirects blood flow to the muscles, and elevates blood sugar levels, providing the necessary energy to face the threat. Fear triggers a response that can manifest in 3 main ways: flight, fight, or freeze. The amygdala communicates with various regions of the brain that control these responses, including the hypothalamus, periaqueductal gray, PAG, area, and locus coeruleus. Each of these areas plays a specific role in orchestrating the body's reaction to fear. When the amygdala signals the need to flee, the hypothalamus activates motor systems that prepare the muscles for a quick escape. The fight response, on the other hand, involves mobilizing physical resources to confront the threat head-on. The locus coeruleus, responsible for norepinephrine release, amplifies the state of alertness and reaction capacity. Finally, the freeze response is an evolutionary strategy that can confuse the predator, making the prey temporarily invisible. This reaction is mediated by the periaqueductal gray area, which regulates tonic immobility. Understanding these neural circuits reveals the complexity and sophistication of the human brain. The amygdala, though small, acts as a conductor, coordinating a symphony of responses involving multiple areas of the brain and body. Its ability to integrate sensory and emotional information is fundamental to survival. However, this same capability can become a source of suffering when the system is hyperactivated, as occurs in cases of trauma and PTSD. Studying the neural circuits of fear also highlights the importance of therapeutic interventions aimed at regulating the activity of the amygdala and associated systems. Behavioral therapies, such as gradual exposure to feared stimuli, can help reconfigure these neural networks, reducing the intensity of fear responses. Medications that modulate HPA axis activity or block norepinephrine effects have also shown promise in treating anxiety disorders and PTSD. The relationship between the amygdala and other brain areas is not one-dimensional, it is a complex network of interactions that evolve based on experiences and contexts. For example, the hippocampus, involved in memory formation, can influence how the amygdala responds to future threats. Traumatic memories stored in the hippocampus can intensify the amygdala's response to stimuli that would otherwise be harmless. This reactivation cycle is a central feature of PTSD, where the mind becomes a prisoner of painful memories. Additionally, the prefrontal cortex, responsible for reasoning and decision-making, can exert inhibitory control over the amygdala. In normal situations, this control helps moderate the fear response by rationally assessing the threat level. However, in individuals with PTSD, this modulation may be compromised, resulting in a disproportionate response to danger. Current research explores ways to strengthen this communication between the prefrontal cortex and the amygdala. Techniques such as exposure therapy, combined with mindfulness practices, have shown efficacy in restoring the balance between reason and emotion. Neuroplasticity, the brain's ability to reorganize its connections, offers a window of hope for reversing the detrimental effects of trauma. Even more revolutionary are brain-machine interfaces, which aim to alter neural activity directly. These emerging technologies, although still in the early stages, promise precise interventions that can deactivate or modify specific circuits involved in the fear response. Imagine a future where implantable devices can monitor and adjust amygdala activity in real time, providing immediate relief to individuals with PTSD. Understanding the neural circuits of fear is more than an academic exploration, it is a journey to understand the very fabric of human experience. Each discovery brings us closer to solutions that can transform lives, offering hope and relief to those living in the shadow of fear. However, this field of study also reminds us of the fragility and complexity of the human mind, inviting us to approach the topic with sensitivity and respect. Chapter 3: Learning and Extinction of Fear. Imagine a scene where a familiar, everyday sound suddenly becomes a warning of imminent danger. This is the heart of classical conditioning, a process through which fears are learned through associations. Pioneering experiments by Ivan Pavlov, initially investigating salivary responses in dogs, revealed a fundamental truth about how living beings can associate a neutral stimulus with a reflexive response. If a sound is repeatedly paired with an unpleasant experience, like an electric shock, the sound alone can eventually evoke a fear response, even in the absence of the shock. This phenomenon is not limited to Pavlov's dogs. It is a demonstration of how our brains are programmed to associate experiences and respond to them. The learning of fear is a process that goes beyond simple associations. It involves physical changes in the brain, specifically at the synapses, the junctions where neurons communicate. This process is known as long-term potentiation, LTP. LTP is essentially a form of synaptic reinforcement, where connections between neurons are strengthened with repetition, making signal transmission more efficient. In the context of fear, this means that fear experiences become engraved in neural structure, strengthening the response to future similar stimuli. The NMDA receptor, a protein present at synapses, plays a crucial role in LTP. It acts as a gateway that allows the passage of calcium and sodium ions when activated, facilitating changes that strengthen synaptic connections. This molecular mechanism is fundamental to learning and memory, including fear memories. When a fear experience is repeated, NMDA receptors at specific synapses are repeatedly activated, leading to a persistent strengthening of these connections. Understanding the learning of fear is just one part of the equation. The other part is understanding how fear can be unlearned or extinguished. Fear extinction is a fascinating process that involves weakening previously strengthened neural connections. However, extinction is not simply erasing a fear memory. It is a learning process that requires the replacement of fear memories with new positive experiences. This replacement process requires the creation of new synapses that compete with the old ones. When a person is repeatedly exposed to a stimulus that once caused fear but in a safe context, their brain begins to form new associations. For example, if someone fears dogs due to a traumatic experience, gradual and controlled exposure to friendly dogs can help form new memories that associate dogs with safety and comfort, rather than danger. Fear extinction is a clear example of brain plasticity, the brain's ability to reorganize and form new connections throughout life. This ability is the basis of many therapeutic interventions for anxiety disorders and PTSD. Exposure therapy, where individuals are gradually exposed to feared stimuli in a controlled and safe environment, is a practical application of fear extinction. This approach leverages the brain's plasticity to help replace fear memories with new memories that do not evoke the same intense emotional response. At the neurobiological level, fear extinction also involves a series of molecular and cellular processes. The prefrontal cortex, a brain region involved in planning and decision-making, plays an important role in modulating the fear response. This area can inhibit amygdala activity, helping to reduce the emotional response to the feared stimulus. Additionally, the hippocampus, which is crucial for forming contextual memories, helps distinguish between safe and dangerous situations, facilitating fear extinction. Another intriguing aspect of fear extinction is its relationship with time. Fear memories, especially those formed during traumatic events, can be particularly persistent and resistant to extinction. However, recent research suggests that there are windows of opportunity, periods during which memories are more susceptible to modification. These periods can be leveraged for more effective therapeutic interventions. The combination of behavioral therapies with pharmacological interventions offers new possibilities for treating fear-related disorders. Drugs that modulate NMDA receptor activity, for example, can enhance the extinction process, facilitating the formation of new memories that compete with the old ones. Additionally, brain stimulation techniques, such as transcranial magnetic stimulation, are being explored as means to directly alter neural activity in specific brain regions, providing additional relief for those suffering from PTSD and other anxiety disorders. However, fear extinction is not a process that happens overnight. It requires repetition and patience from both the individual and healthcare professionals. The human mind, with all its complexities, has a remarkable capacity for adaptation, but this adaptation takes time and effort. A deep understanding of the mechanisms behind fear learning and extinction is essential for developing more effective and personalized interventions. The journey to unlearn fear is, in many ways, a rediscovery of the brain's innate capacity for resilience. It is a journey through synapses, where each strengthened or weakened connection tells a story of adaptation and survival. As we explore these neural pathways, we reveal not only the mechanisms of our suffering but also the keys to our recovery. In a broader sense, the study of fear learning and extinction teaches us about the plasticity of human experience. Every memory, every emotional response is a testament to the brain's capacity to shape and transform in response to the environment. This plasticity is the foundation of our ability to change, grow, and overcome challenges. It reminds us that even in the depths of fear and trauma, there is an inherent potential for healing and renewal. Chapter 4: Therapies for Fear and Trauma. Imagine being trapped in an endless cycle of anxiety, where every small sound, every slight change in the environment triggers an overwhelming fear reaction. For many living with anxiety disorders and PTSD, this is a daily reality. However, science and psychology have developed various therapeutic approaches to help break this cycle. Among them, behavioral therapies stand out for their effectiveness and depth of impact. Prolonged exposure therapy is one of the most studied and applied approaches. In this technique, the patient is gradually exposed to traumatic memories in a safe and controlled environment. By repeatedly revisiting the trauma, the anxiety response begins to diminish. The brain, in its plasticity, learns to dissociate the stimulus from the intense fear response, facilitating the extinction of fear memories. The process is not easy and can be emotionally exhausting, but the results show a significant reduction in PTSD symptoms. Cognitive processing therapy, a variation of cognitive behavioral therapy, CBT, offers another intervention path. Focused on restructuring the thoughts and beliefs that arise after trauma, this method helps individuals develop a more adaptive and less threatening understanding of their experiences. By challenging and modifying dysfunctional beliefs, patients can reduce the intensity of their emotional responses to trauma. This approach not only alters the perception of the traumatic event but also improves the ability to face future challenges with greater resilience. Another innovative technique is eye movement desensitization and reprocessing, EMDR. This therapy involves moving the eyes from side to side while recalling the trauma. The idea is that these eye movements help process and integrate traumatic memories in a way that reduces the activation of threat circuits. EMDR has shown promising results, especially in patients who have not responded well to other forms of therapy. Although the exact mechanism of action is still under study, clinical practice has demonstrated its effectiveness in reducing PTSD symptoms. Social connection also plays a crucial role in regulating fear and anxiety responses. Studies reveal that social interaction can reduce levels of tachykinin, a molecule associated with anxiety and irritability. On the other hand, social isolation can increase levels of this substance, exacerbating anxiety symptoms. The sense of belonging and social support can act as a powerful antidote to fear, promoting an environment of safety and trust that facilitates recovery. The impact of social connection goes beyond simple human contact. Interaction with other humans activates brain areas that promote a sense of well-being and reduce the perception of threat. At a molecular level, tachykinin is a neuropeptide involved in modulating stress and emotional responses. Elevated levels of this molecule are associated with feelings of anxiety and irritability. However, the presence of positive social interactions can significantly reduce these levels, providing a calming and stabilizing effect. Social isolation, on the other hand, has the opposite effect. The absence of connection with others can lead to an increase in tachykinin levels, amplifying feelings of anxiety and stress response. This dynamic reveals the crucial importance of social networks in maintaining mental health. It's not just about having company, but about participating in interactions that promote a sense of belonging and emotional support. Therefore, the therapeutic environment should not be seen only as a space for individual treatment but also as a context that values and promotes human interconnection. Mental health professionals often encourage their patients to seek and strengthen their social networks, understanding that the recovery from deep trauma is a collective effort. Integrating these social dimensions into the therapeutic process can amplify the beneficial effects of traditional interventions, creating a virtuous circle of healing and mutual support. The impact of behavioral therapies, such as prolonged exposure and cognitive processing, is reinforced by the underlying biology of fear and its extinction. The brain, in its adaptive capacity, can learn to respond in less intense and more controlled ways to previously threatening stimuli. This capacity to reshape and reconfigure emotional responses is a testament to the inherent resilience of human beings. However, it is important to remember that the therapeutic process is highly individualized. What works for one patient may not be effective for another. Personalizing therapeutic approaches is crucial to maximize benefits and meet the specific needs of each individual. This may involve a combination of different techniques, adjusted and adapted over time to track the patient's progress. Advances in understanding therapies for fear and trauma not only expand the available treatment options but also offer hope for those who suffer. Science continues to reveal new ways to address fear, from behavioral interventions to advances in pharmacology and emerging technologies. Each new discovery represents a step forward in the fight against the debilitating effects of trauma. Chapter 5: Transgenerational Trauma. Imagine a scenario where the shadows of the past not only shape the present but also cast a deep and lasting influence on future generations. This is the essence of transgenerational trauma, a phenomenon that challenges the notion that individual suffering ends with the person experiencing it. Instead, trauma can be a silent inheritance, transmitted from one generation to the next through complex biological mechanisms. The idea that traumatic experiences can be genetically transmitted may seem like something out of science fiction, but science is beginning to unravel the intricate threads that link the past to the present in surprising ways. Epigenetic modifications, which are changes in gene expression without altering the DNA sequence, play a crucial role in this process. These changes can be induced by traumatic events and, remarkably, can be passed on to offspring. One of the systems most affected by these modifications is the glucocorticoid axis, particularly the regulation of cortisol, the stress hormone. Severe trauma can lead to lasting changes in how the body regulates and responds to cortisol, an effect that can be observed not only in the directly affected individual but also in their descendants. These changes can predispose subsequent generations to a more intense stress response and a greater vulnerability to trauma-related disorders. Animal studies have provided valuable insights into how these epigenetic modifications work. For example, rats exposed to extreme stress during gestation produce offspring that exhibit behavioral and physiological stress alterations. These alterations are mediated by changes in DNA methylation at specific genes related to the stress response. When these modifications are transmitted, the offspring display an amplified stress response, even without direct exposure to the original traumatic event. In humans, evidence is beginning to emerge that historical traumas, such as the Holocaust or slavery, may have left biological marks on the descendants of the victims. Research indicates that children of Holocaust survivors, for example, have altered cortisol levels and are at greater risk of developing anxiety and depression disorders. These findings suggest that the impact of trauma is not limited to the experienced event but can reverberate through generations, shaping the biology and psychology of individuals who did not directly experience the traumatic event. Investigating transgenerational trauma is not limited to biochemical changes. The social and cultural context also plays a significant role. Families living in the shadow of historical traumas often transmit narratives of fear, distrust, and insecurity, perpetuating a cycle of stress and anxiety. These cultural legacies can reinforce epigenetic changes, creating an environment where trauma becomes an intrinsic part of collective identity. Understanding these mechanisms opens doors to new therapeutic approaches. If trauma can be transmitted from generation to generation, interventions may need to address not only the individual but also family and community dynamics. Therapies that focus on family narratives and reinterpreting traumatic experiences can help break the cycle of trauma transmission. Additionally, strategies that promote resilience and well-being at the community level can mitigate the effects of historical trauma, creating a healthier environment for future generations. Research on transgenerational trauma also raises profound ethical and philosophical questions. If past traumas can influence the biology of future generations, how should we address collective responsibility for historical suffering? And how can we use this knowledge to promote a more just and empathetic society? These questions have no easy answers, but they highlight the importance of a holistic and interdisciplinary approach to investigating and treating trauma. Beyond traditional therapeutic interventions, science is increasingly exploring the possibility of epigenetic interventions. As our understanding of epigenetic modifications deepens, opportunities arise to develop therapies that can reverse or mitigate trauma-induced changes. These innovative approaches offer hope for breaking the cycle of transmitted trauma, allowing individuals and communities to free themselves from the chains of a painful past. The multifaceted nature of transgenerational trauma requires us to address the problem from multiple fronts. It is essential to consider the biological, psychological, and social aspects when developing intervention strategies. Collaboration among scientists, clinicians, community leaders, and policymakers is crucial to creating an environment where the impact of trauma can be effectively addressed and mitigated. Chapter 6: Emerging Pharmacological Treatments. In the depths of psychopharmacological science, new horizons are beginning to unfold, bringing promises of transformation in trauma treatment. Among the most intriguing and innovative approaches are therapies assisted by substances like ketamine and MDMA. These drugs, once seen exclusively as recreational or anesthetic, are now emerging as powerful tools in the arsenal against PTSD and other trauma-related conditions. Ketamine, a dissociative anesthetic, has been gaining prominence for its ability to help patients reprocess trauma uniquely. Unlike other traditional pharmacological therapies, ketamine does not simply alleviate anxiety and depression symptoms. It appears to have the ability to reconfigure how the brain processes traumatic memories. This substance can induce a state of dissociation, where patients feel disconnected from their bodies and emotions, allowing a new perspective on past experiences. One of the underlying mechanisms of ketamine's efficacy is its influence on cortical rhythmic activity. This phenomenon involves changes in brainwave patterns, especially in theta and delta frequencies, associated with states of dissociation and deep emotional processing. During a ketamine-assisted therapy session, altering these rhythms can create a mental space where traumatic memories can be revisited without the intense emotional burden that usually accompanies them. This allows patients to confront and integrate these memories in a healthier and more constructive way. Moreover, ketamine has shown rapid and significant effects in reducing depression and anxiety symptoms, often associated with trauma. These rapid effects are particularly important, as they offer almost immediate relief, something crucial for individuals suffering from acute emotional distress. The combination of rapid symptomatic relief and the possibility of reprocessing traumatic memories makes ketamine a valuable tool in treating PTSD. Another substance that has shown remarkable promise is MDMA, also known as ecstasy in recreational contexts. MDMA-assisted therapy explores this substance's unique properties, which increase dopamine and serotonin levels in the brain. These chemical changes induce intense feelings of connection, empathy, and safety, creating a psychological environment conducive to therapeutic work. During MDMA-assisted therapy sessions, patients often report an increased ability to confront and process traumatic memories. MDMA appears to facilitate rewriting painful memories, allowing them to be integrated more healthily into the individual's psyche. The emotional security the drug provides is crucial for this process, as it reduces the resistance and fear associated with confronting traumatic experiences. In addition to increasing dopamine and serotonin levels, MDMA also affects the amygdala, the brain part associated with fear processing. Amygdala activity tends to decrease under MDMA's influence, reducing fear and anxiety responses. This combination of neurochemical and psychological effects creates a unique environment where the extinction of traumatic memories can occur more effectively and quickly. MDMA-assisted therapy is being rigorously studied in clinical trials and has shown promising results, especially for patients who have not responded well to other forms of treatment. The United States Food and Drug Administration, FDA, has granted MDMA-assisted therapy the designation of breakthrough therapy, a status that accelerates the review and approval process, given its potential to offer significant benefits over existing therapies. While these emerging pharmacological therapies offer new hopes, they also require a careful and well-structured approach. Administering ketamine and MDMA in therapeutic contexts must be conducted by trained professionals in controlled environments, ensuring patients' safety and well-being. The therapeutic environment, prior preparation, and post-session follow-up are essential components to maximize benefits and minimize risks. Ongoing research on ketamine and MDMA reveals not only the mechanisms by which these substances act but also highlights the complexity of trauma and the need for multifaceted approaches. Trauma, with its deep roots and extensive ramifications, often requires interventions that go beyond conventional therapies. Introducing these psychedelic substances into the therapeutic field represents a bold move to explore uncharted territories, hoping to find new ways to heal. Beyond the immediate effects and possibilities of trauma reprocessing, these therapies also offer insights into the plasticity of the human brain. The brain's ability to adapt, form new connections, and reconfigure emotional responses is a testament to the resilience inherent in the human condition. By using substances like ketamine and MDMA, mental health professionals are not only treating symptoms but also facilitating deep transformation processes. Exploring these emerging treatments leads us to reflect on the future of psychotherapy and psychiatry. As we continue to unravel the mind's mysteries and discover new ways to promote healing, we are challenged to reconsider our traditional approaches and embrace innovations that, although initially controversial, can offer a new dawn in trauma therapy. Chapter 7: Assessing and Self-Managing Fear and Trauma. Fear, a primal and deeply ingrained emotion, is not just an automatic response to external stimuli but also a complex construct shaped by the interaction between our internal perception and external events. Understanding this dynamic is crucial for assessing and self-managing fear and trauma, and here, the distinction between interoceptive and exteroceptive perception becomes fundamental. Interoceptive perception refers to the ability to sense and interpret internal body signals, such as heartbeat, muscle tension, and visceral sensations. This ability allows us to be aware of our own physical responses to fear and stress. Exteroceptive perception, on the other hand, involves detecting external stimuli, such as sounds, sights, and smells, that can be perceived as threats. How we integrate these two forms of perception determines our overall response to fear. The insular cortex, a brain region often overlooked in popular neuroscience discussions, plays a crucial role in calibrating the intensity of our internal responses to external stimuli. This brain nucleus acts as an intermediary, helping align our internal sensations with external realities. When functioning well, the insular cortex allows us to respond proportionally to perceived danger, avoiding both exaggerated and underestimated reactions. However, in cases of trauma or chronic stress, this calibration can become misaligned. The brain can become hypersensitive to external stimuli or, conversely, lose the ability to recognize important internal signals. For example, a person who has suffered trauma may experience a significant increase in interoceptive perception, where elevated heartbeats or accelerated breathing are constantly interpreted as signs of imminent danger, even in the absence of external threats. This mismatch between internal and external perception significantly contributes to maintaining and intensifying anxiety and PTSD symptoms. Accurately assessing the internal response relative to external events is a crucial step for self-managing fear and trauma. Techniques such as mindfulness meditation and biofeedback are powerful tools that help realign this perception. Mindfulness, by training the mind to focus on the present and observe internal sensations without judgment, can reduce hypervigilance and emotional reactivity. Biofeedback, in turn, uses technological devices to provide real-time information about physiological processes, allowing individuals to learn to consciously control physical stress responses. Another fascinating and counterintuitive aspect of fear control involves deliberate and controlled exposure to stress. Recent research suggests that subjecting oneself to short periods of deliberate stress can have profound therapeutic effects. This concept, which at first glance may seem paradoxical, is based on the idea that controlled stress exposure can help recalibrate the body's stress response system. By facing stressors in a controlled environment, the brain and body learn to better manage and regulate stress responses. This approach can help desensitize the nervous system, decreasing the intensity of stress and fear responses in future situations. Deliberate stress exposure, therefore, not only strengthens resilience but can also reduce symptoms of depression and chronic fear. Practices such as sauna, ice baths, and intense exercise are examples of methods that induce deliberate stress. During these activities, the body is subjected to physical stressors that activate the stress response system, but in a controlled and temporary way. By overcoming these experiences, individuals not only improve their physical capacity but also strengthen mental and emotional resilience. The idea that short-term stress exposure can be beneficial is rooted in the concept of hormesis, where small doses of a stressor can strengthen the organism. In the context of fear and trauma control, hormesis suggests that repeated and controlled exposure to stressors can reconfigure the stress response, promoting a balance between interoceptive and exteroceptive perception. Balancing these two forms of perception is essential for an adaptive fear response. When this balance is achieved, we can respond to challenges proportionally and effectively, without being dominated by automatic and disproportionate responses. Developing this balance requires continuous and intentional practices that cultivate internal awareness and external resilience. Beyond individual practices, integrating therapeutic approaches that incorporate stress exposure and perceptual recalibration can be extremely beneficial. Therapies such as acceptance and commitment therapy, ACT, and dialectical behavior therapy, DBT, use techniques that help individuals accept their internal experiences and engage in behaviors that promote resilience. ACT, for example, emphasizes accepting internal experiences while focusing on actions aligned with personal values. This approach helps individuals develop a healthier relationship with their thoughts and emotions, allowing a more adaptive stress response. DBT, on the other hand, combines acceptance and change techniques, using mindfulness, emotional regulation, and distress tolerance skills to help individuals navigate and overcome emotional crises. Exploring these approaches makes it evident that fear and trauma control is neither a simple nor a linear task. It requires a deep understanding of the interactions between our internal perceptions and external stimuli, as well as the willingness to engage in practices that challenge and expand our stress response capacity. The journey of self-managing fear is a continuous process of learning, adaptation, and growth. Science continues to reveal the complexity of the fear response, highlighting the importance of an integrated approach that combines self-awareness practices, controlled stress exposure, and therapeutic interventions. Each of these practices contributes to building a solid foundation of resilience and emotional control, allowing individuals to navigate adversities with greater confidence and effectiveness. Exploring these dimensions of fear and trauma response invites us to reflect on the inherent resilience of the human being. Our capacity to adapt, learn, and overcome is deeply rooted in our biology and psychology. By developing practices that cultivate this resilience, we can not only face immediate challenges but also build a foundation of strength and stability that supports us throughout life. Chapter 8: General Support and Supplementation. As we explore the vast territories of mental health, we find a tangle of factors that influence our ability to deal with stress and trauma. Among these factors, lifestyle aspects stand out, which, although often underestimated, play a crucial role in maintaining emotional balance and promoting well-being. Adequate nutrition, regular sleep, and social connection are not merely complements to treatment but fundamental pillars that support mental health. Nutrition, at its core, is more than just providing energy to the body, it is a powerful tool that can directly influence brain function. Foods rich in essential nutrients, such as omega-3 fatty acids, B vitamins, and antioxidants, have demonstrated beneficial effects in reducing brain inflammation and promoting mental health. A balanced diet, rich in fruits, vegetables, lean proteins, and healthy fats, can help stabilize mood and reduce anxiety. Sleep, in turn, is a restorative state that allows the brain to process experiences and consolidate memories. Sleep deprivation can exacerbate stress and trauma symptoms, while restorative sleep can improve emotional resilience. Establishing a consistent sleep routine, avoiding stimulants like caffeine and blue light from screens before bed, can promote better sleep quality and, consequently, better mental health. Social connection, a recurring theme in the discussion of emotional support, is another vital component. Interaction with others, whether through friends, family, or support groups, provides a sense of belonging and validation. Studies have shown that the lack of social connection is associated with higher levels of tachykinins, molecules that can increase anxiety and irritability. Maintaining meaningful relationships and seeking social support can, therefore, act as a powerful antidote to the debilitating effects of stress and trauma. Beyond these lifestyle factors, supplementation can offer additional chemical support for stress reduction and mental well-being promotion. Among the most studied supplements are saffron, inositol, and kava, each with its unique properties and potential benefits. Saffron, a spice derived from the crocus sativus flower, has been used for centuries in traditional medicine to treat a variety of conditions. Modern research has begun to unveil its positive effects on mood and anxiety. Active compounds in saffron, such as crocin and safranal, have demonstrated antidepressant and anxiolytic properties, possibly through modulating serotonin levels in the brain. Supplementation with saffron can help alleviate depression and anxiety symptoms, offering a natural approach to emotional support. Inositol, a substance naturally found in many foods and also available as a supplement, plays a crucial role in cell signaling and mood regulation. Studies indicate that inositol may be effective in treating anxiety and depression disorders, especially in cases of panic disorder and obsessive-compulsive disorder. The exact mechanism by which inositol exerts its effects is not fully understood, but it is believed to be related to its ability to improve serotonin signaling in the brain. Kava, a plant native to the Pacific Islands, is known for its relaxing and anxiolytic properties. The kava root contains compounds called kavalactones, which interact with GABA receptors in the brain, promoting a calming effect without the sedative effects associated with many pharmaceutical anxiolytics. Supplementation with kava can be an effective option for reducing stress and anxiety, providing natural and safe relief. While exploring the benefits of these supplements, it is important to remember that the approach to mental health must be holistic and personalized. What works for one person may not be effective for another, and the integration of supplements must be done with care and professional guidance. Combining a balanced diet, adequate sleep, meaningful social connections, and appropriate supplementation can create a robust foundation for emotional resilience and mental well-being. The quest for emotional balance and managing stress and trauma is a complex journey requiring a multifaceted approach. Integrating lifestyle factors and supplementation not only complements other forms of treatment but also strengthens the body's inherent ability to heal and adapt. By taking care of the body and mind comprehensively, we can create an internal environment that favors mental and emotional health, allowing us to face life's challenges with greater strength and resilience. In this book, we explored the complexity of the neuroscience of fear and trauma, from the neural circuits involved to modern therapeutic interventions. We understand that fear is a fundamental emotion regulated by complex biological systems and that trauma can be genetically transmitted, impacting generations. We discussed various therapeutic approaches, such as prolonged exposure therapy and therapies assisted by ketamine and MDMA, offering new paths to healing. Natural supplements like saffron, inositol, and kava can complement these approaches. We also emphasize the importance of lifestyle factors, nutrition, sleep, and social connection, as essential pillars for mental health. These elements help strengthen our resilience and adaptability. By integrating these approaches and understanding the multiple facets of fear and trauma, we can adopt practices that not only treat symptoms but also promote emotional and mental health. This holistic knowledge empowers us to transform fear and trauma into opportunities for personal growth and resilience, offering a path to a more balanced and healthy future.